Okay. Okay, yeah. So the title of my talk is the same as the name of the workshop, Mathematical Modeling in the Machine Learning Era. So I will try to say something about mathematical modeling in the relation to machine learning. And I will do this by talking about a project that has been done within this larger SSF project. And the title of that project is Quantifying Cell Line Specific Proliferation and Migration Rates in Glioblastoma Cells. And it's a work that has been carried out jointly with the Nylander lab in Uppsala, in particular Sven Nylander and a PhD student, Emil Rosén. So this project aims at understanding differences in cancer cells that come from different patients, and glioblastoma is a brain tumor. I will talk a bit more about that on the next slide. So I will talk a bit about the data and the method that we've used, and then say something about some results that we have. And then I will sort of come back and zoom out and talk a bit about the development of mathematical biology, which is my field of mathematical modeling, and do this in the light of machine learning and statistics. Okay, so moving on. So glioblastoma, or GBM for short, is the most common primary brain tumor that we find in adults. Prognosis for these patients is very poor, approximately 1 year average survival after diagnosis. And this has not improved much in the last decades. The only improvement that we've seen is a new drug, temozolomide, which was discovered, I think, roughly 20 years ago, that prolongs survival by 3 months on average. So there's much to be done here. But it's a difficult task, and there are several challenges here for the treatment of glioblastomas. So when it comes to drugs, for example, drug delivery is difficult because we're delivering the drug to a sensitive organ with a very specialized vasculature that will only let certain molecules in. In terms of surgery, treatment is difficult because this tumor grows in a very diffuse manner with an infiltrative morphology, where cancer cells tend to migrate into the healthy tissue, which makes it difficult to find the border between tumor and healthy tissue. And this means that when the tumor is surgically removed, there is always cancer cells left behind that give rise to a recurring tumor. Then we also have the problem of a high level of heterogeneity. So there, these patients, the molecular makeup of these tumors is very different. They have very different mutations, and therefore different pathways that are upregulated. And this means that it's not easy to define a single target for these patients. So what has then been done here? Well, the Nylander lab, together with AstraZeneca, carried out a large-scale drug screen of these glioblastoma cells. So they were looking for drugs that had an effect on reducing proliferation, so reducing cell division or even killing cells. And this was done on a very large scale. So they, in Uppsala, have collected samples, biopsies from many different patients, and here they used cells that come from 100 different patients. On these 100 cells, they tried 248 different compounds, drugs, and they did so under 22 separate doses. So what it looks like, the sort of the outcome here is illustrated here in these 3 plots for 3 different drugs. So what we have on the x-axis is the dose of the drug, and on the y-axis we have surviving fraction of cancer cells. And here yellow and blue correspond to different cell lines, and all the different lines corresponds to different replicates that have been carried out. So we see, in this case, we see a very different response to the same drug between these 2 different cell lines. So So this is a lot of data that has been gathered in this drug screen. And they initially only looked at the effects of proliferation. But as I said before, these cells tend to migrate into healthy tissue, and therefore changing cell migration with drugs could also be an interesting way of treating these patients, because that would make the tumor more compact and easier to remove surgically, for example. So what we have done is to try to see if is it possible to say something about cell migration based on this data available. And it actually is, because what is actually being measured in this drug screen is the data is basically images that were taken of the cells after 96 hours of drug exposure. So a typical image in the drug will look in the screen will look like this, and then we have one image for each separate experiment. So as you see, these two images differ quite markedly. Here we have cells sort of evenly spread out, and here they're lumped together. And we can actually, I'll explain this in a second, this actually sort of bears a signature of how much the cells have migrated in the experiment. And we quantify this migration, or the pattern that we see, using a pair correlation function. The pair correlation function measures the normalized density of cell pairs that are separated by a certain distance r. And it's normalized in the way that if we don't have any structure or any correlations, spatial correlations, this pair correlation function will take the value of 1. So if we measure the pair correlation function for these two images, then this left image here, which is shown in blue here, has a pair correlation function which goes from 0 because the cells can't overlap, so we won't have any pairs at the distance 0. And then very rapidly goes on up to a value of approximately 1. In this case here, where cells are lumped together, we see a very clear peak in the pair correlation function, which means that we have sort of over-representation of cell pairs that are at a distance of 20 microns, roughly the diameter of a cell here. Okay, so this is how we will quantify these spatial correlations. We then used a model, a cell-based model of this drug screen assay to investigate how cell migration affects this pair correlation function. So we mimic the initial condition of the experiment by seeding cells randomly in a square region, a 2D region, representing the well in which the cells are growing. And we have 1,000 cells per well, which is the same as the experimental protocol. Now, this cell population, the dynamics of it are driven by random migration, so the cells will move about randomly. But they will also bump into one another and experience intracellular forces. So this means that the motion of each cell can be described by a stochastic differential equation, which captures intracellular forces and a diffusion term here, or a random walk. So this will govern the motion of each cell. And then lastly, the cells will also divide with a certain rate alpha. Okay, so that is our model, and I'll briefly show how it looks like. So in the left panel here, we have cells with a low migration rate. Cells are wiggling about a bit, they are dividing, and we see the formation of these lumps or sort of islands of cells. So this is typically what it will look like when we have a low migration rate or low diffusion. Whereas here we have a high migration rate, and we see that cells are wiggling about a lot more, and they're not forming these lumps. Rather, it looks like that we have a more uniform distribution in space of cells. And if we then use our measure of spatial correlations, the pair correlation function, we see that this looks very different for these two simulations. So the simulation with a high migration rate, as expected, the pair correlation function rapidly goes up to 1 and then stays there, meaning that we don't have any spatial correlations. Whereas for the case of low migration rate, we have this peak. And and then a slow decline in the pair correlation function. So this we will use in order to calibrate the model and find and infer the parameters for each experiment. So how do we do this? Well, we use approximate Bayesian computation, which is a simulation-based tool for inference. I will not go into that much detail, but essentially what we do is that we infer for each image the migration rate D and the baseline proliferation rate alpha. In order to do this, we need somehow to compare data and simulation output. And we do that with what is known as summary statistics. And we pick two of those. The cell count, simply the number of cells in the well and in the simulation. And then we also compare the pair correlation function from the simulation and from the data. And given a prior distribution, this ABC algorithm provides us with a posterior distribution of parameter values when we compare the model to the data. Okay, so that's the approach that we're using, and I just wanted to sort of show it in a more schematic way here, how everything sort of fits together. So we have our agent-based model on the left here. We want to compare it and parameterize it using this data. And the ABC algorithm here sits sort of in between the agent-based model and the data. By comparing or forming summary statistics from the data and the model, we can then accept or reject certain parameter values. And in the end, that will give us a posterior distribution of parameter values. Now, and I will come to this in a second, because we're dealing with such a large data set, roughly 10 to the 5 different experimental conditions, then we will get very many parameter values. And to try to make sense of those parameter values, we then apply a mixed effects model where we try to separate out cell-specific effects on the parameters and drug-specific effects. And I will come to this in a second. And I will come back to this towards the end of the talk. But what the point here is that this sort of agent-based model sits within a much larger framework of methods and techniques that come from machine learning and statistics. So one result that we noticed is that when we look at a negative control well, so this is experiments where no drug is added, the cells are just growing without any perturbation, then what we see is a correlation between diffusion or migration rate of the cells and the doubling time of the cells. So cells that have a short doubling time, so they divide very quickly, they tend to have a low migration rate, whereas the opposite is true. So cells that have a long doubling time, they divide slowly, they tend to move more. And this has been observed before in glioblastomas. It's called the go versus grow trade-off. In terms of the data that contains experiments with drugs, then we, as I said before, we use this ABC to infer for each drug dose and cell line combination a pair of parameters, alpha, the proliferation rate, and D, the diffusion coefficient. And we have roughly 10 to the 5 of those pairs. So that's a lot. And to analyze the parameter values, we use a mixed effects modeling framework. So the way this is done is the following. So here Dij of X is the diffusion constant for cell line I, of which we have approximately 100, and drug J, we have approximately 200. At a drug dose X, which is the logarithm of 1 plus the dose. So this diffusion coefficient or constant, we can then separate out into a baseline diffusion D naught, which is the same for all experiments. We have a shared dose effect, which is drug specific, but shared by all cell lines. And then here we have a cell line specific effect, which is basically like a constant diffusion coefficient for the cell line, and then a cell line and drug specific effect, Cij here. And then we also have an error term. And this we do exactly the same way for the proliferation rate. And now if we look at these shared effects and plot them against each other, we see what appears as a negative correlation between effects on proliferation and migration. So this means that if we have a drug such as this one, which reduces the proliferation rate, then it tends to increase the migration rate. And the reason why we have a majority of data here on the left hand side is because the drugs typically tend to reduce the proliferation rate. Because that's the criteria for them being chosen. So we have a tendency, but we see a clear, we see a correlation here, and this is not good news, because it means that if we have a drug that reduces proliferation, then the cells migrate more and the tumor becomes more widespread in the brain. So not good. But these are only preliminary results so far, and we will validate them using time course data, which is being collected in the Nylander lab, where we actually take images of the cells every hour instead of just a single image. Okay, so that's the project that we've been working on, and I wanted to say something about mathematical biology and data and machine learning. So from my perspective, at least, I think that the relation to data has changed significantly within the field of mathematical biology in the last decades. There is a much larger awareness that the models need to be able to use data and also in a sensible way. So for example, with agent-based models or cell-based models, which we used here, then say a decade or two ago, it was very common that you just looked for a qualitative agreement between model and data. But now, with for example, approximate Bayesian computation, we see more and more actual parametrization of agent-based models. And in terms of parameter estimates, I think that the field has moved forward as well. And also looking back, then just point estimates using some basic least squares methodology was common, but now we see much more work where we use quantification of uncertainty in the model, and this comes with the ABC algorithm, where we get a posterior distribution instead of just a point estimate. And also we see more care taken when it comes to choosing different models, and here Akaike information criterion and Bayesian information criterion comes in, so that we can make say rigorous choices between which model to use instead of just arbitrary ones. So I think that's, at least in my view, how the field has changed. And then maybe concluding here, I'd say I don't think one should view it as a sort of conflict between different methods here, but rather that mathematical modeling should be viewed and used as a cog in a larger machinery. And here we can think of agent-based models or ODEs or PDs that are used together with techniques from machine learning and statistics, just as I've showed in this talk. So those are my thoughts on this. And last night I bumped into or I saw this an advertisement of this paper on Twitter, and I think this is exactly what I'm talking about. So this is a paper with the title "Biologically Informed Neural Networks Guide Mechanistic Modeling from Sparse Experimental Data." And the idea with this paper is that we have in this case a PDE that describes the dynamics of a population of cells that are dividing and migrating, just as in the example I've shown. And what they did is that they represent this PDE using a neural network. and then they can infer functional forms in the PDE using this neural network and training it on data. So I think this is an interesting development, and I think this is what we will see more of in the future. Okay, so that's all for me. So I'll just say something. So the funding, of course, is from the SSF project, and then the Nylander Lab has funding from other places as well, AstraZeneca, KAW, Cancerfonden, and Vetenskapsrådet. Okay, so that's all for me. Thanks a lot.
Thank you, Philip. I think we said before to have discussion after the first two talks, but we have a couple of minutes. So maybe if somebody just raise his or her voice to ask a question, maybe Philip can answer that.
Can I ask a question? Philip, it was very interesting to see this correlation between the proliferation rate and migration rate. Do you have some idea about a hidden factor behind this correlation? So it's not causation relationship. I thought about cancer cell age. Can it be so that it's young, it proliferates, but when he produces some children, now it goes down in the speed?
I mean, there are some ideas about why this happens. So, and one is that the cell migration and cell division are sort of competing processes because they both need the cytoskeleton. So the cell, when it migrates, it has to rearrange its cytoskeleton and create new attachment points and pull itself forward. And when it divides, it also needs its cytoskeleton because that's what it uses to pull apart the nuclei into two daughter cells. So it could be that, this is one hypothesis, is that it's simply a matter of that you can't do both at the same time. So the cell will prioritize one process over the other, basically.
Thanks. Maybe one more question, actually. Please just ask.
Another question, maybe?
Yes, please.
Yeah, just to follow up on the previous question. So would that explain somehow the correlation you see also in the treatment effects? Or why would they start migrating when they are stopped from?
So that could be another thing, and there people have speculated that what is causing this is that the drugs basically are stressing the cells. So they are sensing that they are close to dying, and therefore they're running away, basically. That could be that's one explanation. It's been seen in other types of cancer as well, that some drugs have a tendency to increase migration, and they've traced this to certain pathways that are basically stress response. So that's one possible explanation.
And one other brief one. Did you compare these ABC methods to more traditional, simpler methods, and sort of, is it possible to make a comparison?
Yeah, we didn't do this. So Emil, who's done most of the work here, he was very fond of the ABC. So that's what we've done. But of course, yeah, you could also use other methods.
Thanks again for these answers, and I think we now move on to